Hi everyone, thank you for coming here today. Thank you, Celia, for taking part today. So today I'm going to be speaking about the neural correlates of the therapeutic effects of psychedelics. I know that quite a few of you here don't have a neuroscience background, so I'll try my best to explain perhaps some quite challenging concepts as simply as possible. This is something I've been looking at since the past about 6 years now, since I did my master's at Imperial College London under the supervision of Neil Rosen. So this is quite a surreal experience now lecturing to my previous supervisor. But we're going to give it a go and hopefully, hopefully it works out well. I did my master's at Imperial College, and since then I spent a year working in the psychedelic industry. Some of you might have come to this talk the other week with Henry Fisher. I worked with Clark Health across a number of clinical studies in Spain, and now I'm in my fourth year of my PhD at UCL, where we're doing a big DMT study, and I'll speak about that more so at the end. Let's jump into it by starting with the question, what are psychedelic drugs? You probably know what psychedelic drugs are, that's why you're here. But the term psychedelic comes from the Greek word psyche, meaning soul or mind, and dailun, meaning to manifest. And this is a term coined by Humphrey Osmond in a letter to Aldous Huxley. And over the years, there have been many other words that people have used to describe psychedelics, such as hallucinogen, entheogen, psychomimetic, etc. And this is all trying to capture what the psychedelic experience is. And the word psychedelic is really stuck. So when I say psychedelics, I mean the classic psychedelic, i.e. The serotonergic psychedelic. And we have the phenethylamines, including mescaline, 2CB, and DOI. And mescaline is found in the San Pedro cactus. It's consumed orally, and it lasts around 12 hours. Then we have the tryptamines, such as psilocybin, which is consumed orally, and it lasts around 4 to 6 hours. And then we have DMT, which is consumed also orally in the form of ayahuasca, but you need to take it in combination with a monoamine oxidase inhibitor. Otherwise, it's broken down by the gut and it can't get to the brain. So in my research at UCL, we're using DMT in an intravenous formulation, and this means that it goes straight to the brain. It bypasses metabolism. You can also smoke DMT, and this also goes directly to the brain. You also have 5-MeO-DMT, which is found in the Bufo alvarius toad, and that has a similar phenomenology to DMT, but also slightly different. It's not really considered as one of the classic psychedelics as much. And then we have the ergolines, which is encompassing LSD, and that's consumed orally, lasting around 10 to 12 hours. And then you have some other atypical psychedelics, but they're not really based around this serotonergic structure. So serotonin is a very abundant neurotransmitter in the body and in the brain. It regulates mood and sleep. And you have the phenethylamines here, which have a structure a bit more similar to dopamine, so it has a bit more of a stimulating effect. But again, it's really the mescaline, psilocybin, NMDMT, and LSD, which are really considered these classic psychedelics. And they're being developed for use in psychiatry for a number of different conditions. We also have MDMA, though, which is being developed for PTSD. It's in its phase 3 of research at the moment, as well as ketamine, which I'm sure with Celia in this department, you'll know plenty about. It's being developed for a number of different indications, such as depression, alcohol use disorder, and amongst other things. And then we have ibogaine as well. But what I'm speaking about today.
Is ibogaine a tryptamine or not?
I guess it has quite a messy neuropharmacological activity, but potentially it could be. But yeah, I think it's a bit more messy and so what I'll be speaking about today mostly is the research that's been conducted into the clinical and neuroscientific basis of classic psychedelics. And this isn't new research. So back in the 50s to the 70s, there was a lot of clinical research carried out with psychedelics after LSD was discovered in 1943 by Hoffman. It led to this whole movement of psychedelic research. And in the 50s and 70s, there were 1,000 papers, there were 6 international conferences. The US government spent $4 million to fund 140 studies involving over 40,000 patients. $4 million went a lot further back then than it does now. And this was predominantly LSD research at the time. This was for many psychiatric conditions. Alcohol dependence was a big one. The founder of Alcoholics Anonymous actually attributed a lot of his recovery to LSD. It was also explored for its use in anxiety and terminal illness, in depression, amongst other things. And there were 2 main approaches used back then. You had the psychedelic approach, where you take a low dose of LSD during a session with a psychoanalyst or a psychotherapist, and you do psychoanalysis during the experience. And then you had psychedelic therapy, which is a high dose session where you're not speaking as much. And the work is done more in this preparation integration framework. And this is Stanislav Grof, and he was a psychiatrist that was working on many of the studies back in this period of research. And he tripped at, I think, thousands of people perhaps with LSD psychotherapy. And he wrote in this book, The Realms of the Human Unconscious, that psychedelics used responsibly and with proper caution would be for psychiatry what the microscope is for biology and medicine, and what the telescope is for astronomy. And so it really kind of reflects the power of these drugs to help us in research in neuroscience, but also to help people with psychiatric conditions. And as I just mentioned, psychedelic therapy comes in this sort of container with preparation and integration. So before you even take the drug, you have this period of preparation. And one of my colleagues and friends, Rosalind McAlpine, does a lot of research into the best practices for preparation. And this preparation period includes psychoeducation, so learning about the drug or the medicine that you're about to consume, what you might experience. Also practicing some grounding techniques which might help you navigate these challenging experiences that might arise during the psychedelic trip. Establishing rapport with your therapist, that's a big component of it, building trust and really being able to feel like you can work with this person, as well as setting intentions for what you want to get out of the experience, and that can really help guide what you can get within the psychedelic experience itself. And then you have the psychedelic therapy session itself, where the therapist will take a non-directive and supportive approach. They won't ask probing questions. They won't try and send you down rabbit holes. They'll encourage you to go within. And often they'll wear eye shades and have headphones with a selected music playlist. Sorry. Do a little removal service. They'll be encouraged to really go within and to kind of really, to have the eye shades on and to not actually look outwards, but to focus on what's going on inside. And there'll be a specially selected music playlist often, which really encourages people to take these more emotional journeys and perhaps go to places that they've been avoiding. Hello, yeah.
So when you're talking about this, you did mention that there's some research going on PTSD. Like, did you focus on the specific aspects of PTSD when you did some research? Like embitterment is one of the most predominant is that a PTSD person endures.
So we'll get to more of the sort of mechanisms in a moment.
Maybe if it's okay to save the questions to the end. Is that okay? Is that all right with you?
Yeah.
We can have them throughout. But I don't know. I know your thoughts quite helpful.
Maybe at the end.
Yeah. Is that okay? Thank you.
Then the music really helps people go to particular places that they might be avoiding. And then you have integration. And that's the work done after the trip, where you take the insights and the experiences that you've gained in the acute psychedelic experience and try to bring that to your daily life. And this period can go on for years, perhaps a lifetime. But some studies don't actually use psychedelic therapy, and we'll talk more about that towards the end. But there was that period of early clinical trials, but then all that research came to a halt, and we call it the psychedelic dark ages. From the 70s to the 2000s, there was a lot less research in this space, primarily because of President Nixon's war on drugs. You had the hippies that took the acid from the lab to the festivals, to, out to the streets, and also it was involved in the anti-Vietnam War movement. So all of this led to this political repression of the psychedelic movement. So President Nixon's war on drugs led to the rescheduling of psychedelics to a Schedule 1, which made it a lot more difficult to carry out research in that time. You can see this reflected in this graph here. There was an increase increase around this period in the 70s, and then a bit of a lull. And now in the 2000s plus, we have a real increase in the number of studies published. And now we have this psychedelic renaissance period. So many of you have probably seen the news. It's apparently great for absolutely everything. Here's just some of the headlines. Can psychedelics save the world? Can they? Do we think they can? Anyone?
Yes.
Yeah. Great. Love the enthusiasm. But I think that's all a reflection of the recent clinical research in this space. And I looked it up yesterday on clinicaltrials.gov, which is a registry of all the clinical trials around the world. There are 318 clinical trials currently registered across a number of indications. And by indications, that's a technical term for conditions, really. And this is just a few of them: anxiety, depression, OCD, eating disorders, addiction, cluster headaches, chronic pain, smoking cessation. The list goes on. Pretty much anything that can have psychedelics thrown at it is having psychedelics thrown at it. And there was a meta-analysis published a few years ago that took multiple studies, the results of multiple studies together, and demonstrated that there was a significant robust symptom reduction in the short and medium term with psychedelics compared to placebo, and early indications that this effect might persist into the longer term. But I'm sure over the years, go on, we'll have more evidence into the longer term effect as well. So maybe you've heard a bit about this from Henry Fisher, was it a month ago? Pardon? A week? Since with all this clinical research, a lot of it's been done in the corporadelic industry. I quite like the term corporadelic, means corporate psychedelic. Whether you buy into it or not, that's an interesting point of debate we can maybe chat about at the end. But the medicalization of psychedelics is happening, and this is a map of where all of the psychedelics are in development. And so I don't know how much you know about the pharmaceutical development pipeline, but you have to go through from preclinical research in animal models to phase 1, then phase 2, phase 3, and then it gets to the middle of the circle where it's approval. And in the middle here, you have Janssen's Spravato, which is ketamine nasal spray for treatment-resistant depression. And on this ring out here, you have the phase 3, and that's what you need before you can get it approved into the medical use. And the closest one we have at the moment in terms of psychedelics in this pipeline is Compass Pathways, which are currently in their phase 3 program for psilocybin for treatment-resistant depression. So hopefully in the next few years, it might actually be something that would be that your psychiatrist would be able to prescribe you. And with that, it's been predicted to be worth 18.7 billion by 2023. But what the sort of clinical stuff doesn't really tell you, what the industry doesn't tell you, of how these drugs actually work. And that's kind of why I left industry, to be honest. I was a bit more interested in the mechanisms. And so I'm going to try and give you an overview of the methods we use in neuroscience, also in psychedelic neuroscience, to study how these drugs work. So at the bottom of this upside down hierarchy here, I guess, we have the molecular and cellular level. So we want to look at the receptors, what's going on in the actual cell basis when you introduce psychedelic drugs. And to do this, we use animal models. Quite funny, the animal model we have, but if a mouse is tripping, is just head twitches. So that is our only, I don't know, I had to model that. So that is when you give a mouse a psychedelic drug, it will twitch its head, and the more it's twitching, the higher it is. It's quite difficult to translate the findings from a mouse into a human. Anyway. Then you have in vitro assays where you'll give the drug to a petri dish of cells to look at what's going on in that sort of cell culture. And then we have positron emission tomography, which I'll speak more about in a moment. But in that way, we can actually image this more molecular cellular basis of what's going on the receptor level in humans. You can do it in humans, you can do it in animal models. So it does give a bit more of an understanding of how this molecular cellular basis translates to the higher levels, such as the circuit level. So what's happening on a whole brain level? And this is where most of my research lives, on this hierarchy. To do this, we have techniques such as EEG and fMRI, which I'll again speak more about in a moment. And then at the top of the hierarchy, we have the behavioral and psychological level, which is way more observable. We can ask someone how they're feeling using questionnaires, and we can use behavioral tasks to probe their changes in cognition and mood and a number of things. That's a general overview of the kind of things the kind of methods that we use in psychedelic neuroscience, and we'll kind of go through that over the next hour or so. So on a cellular basis, for those who don't know, this is a synapse, and at the top here we have, does this work? No, it doesn't work. At the top here we have the presynaptic neuron, and then we have the postsynaptic neuron. So what will happen is a signal will come down the presynaptic neuron, and it will lead to the release of a neurotransmitter into the synapse. The serotonin, in this case, will travel across the synapse and bind to these serotonin receptors, and it will lead to a signal in the next neuron, and then it will lead to a chain reaction. But generally speaking, psychedelics stimulate the serotonin 2A receptor. There are a number of serotonin receptors, but it's this 2A receptor that is really key to this, the actions of psychedelics. So it's what we call an agonist. It stimulates the serotonin 2A receptor and leads to this excitatory effect in the postsynaptic neuron. And we know this because if you give someone ketanserin, which is a drug which is what we call an antagonist, it blocks the effects of the serotonin 2A receptor, it will mean that the person will not have any psychedelic effects. We also seen through this cellular and molecular work done in animal models, in mice predominantly, that there is an increase in neuroplasticity after you take a psychedelic. And neuroplasticity is a fundamental process which we all have in our brains, which facilitates learning, for example, and it allows changes in brain connectivity and function. And we see that with psychedelics, it increases this dramatically and rapidly. And we see this by these, so that's before you take a psychedelic, and then after you take a psychedelic, you can see the growth of these synapses. So you have more of these, which lead to changes in brain function overall. And this leads to, this is a baby dressed up as a dinosaur. And there's been evidence that psychedelics basically reopen this critical period. And a critical period is when you're a baby, you don't need to be dressed as a dinosaur, but when you are a baby, you have this critical period where learning is way more easy, and you can rapidly learn and unlearn associations, and it leads to rapid changes in brain organization. And there's evidence that psychedelics increase and potentially reopen this period of neuroplasticity, of this critical period of learning. And this is, interestingly, again, this has been shown in animal work, but they've shown that depending on the length of the drug experience, it dictates how long the critical period is open for. So if you have ketamine, which is much shorter acting, lasts around 30 minutes to 2 hours, the critical period will be open for 48 hours. And then you have LSD, which is longer acting in its acute state. It lasts 8 to 10 hours, and then the critical period is open for 3 weeks. So in that 3-week period, you're way more adaptable. You can learn things way more easily or unlearn them. So that could potentially guide the development of different integration protocols, and can potentially work with that in the psychedelic assisted therapy sort of framework. And then using positron emission tomography, we can see, again, the binding of these receptors in humans and how that relates to the psychedelic experience itself. So with PET, you inject someone with a radioactive tracer, and you have to be selective about the tracer that you're going to use. So in this case, they used a tracer for the serotonin 2A receptor, and they showed that the more psilocin, so the metabolite of psilocybin, the active ingredient, essentially, they show that the more psilocin that's bound to the serotonin 2A receptor, the more intense the psychedelic experience. And I guess this potentially speaks to the microdosing research as well, that, you know, even if you take a small dose of whichever psychedelic, you're still going to get some potential receptor occupancy and potentially this neuroplasticity enhancement. And this has also been shown, this technique has also been used to demonstrate where these serotonin receptors are found. And as you can see from these red dots, that's where the serotonin 2A receptors are found, and it's densely distributed in the cortex, although some more in the subcortical regions down here, which we'll speak more about in a bit. So let's think now about the circuit changes with psychedelics. So what's happening in humans, what's happening on more of a whole brain level. And so this is mostly what I tend to work with, functional magnetic resonance imaging. And so this is detecting changes in blood flow in the brain. We're assuming that if there's a change in blood flow to a certain region, there'll be an increase in demand for oxygen and glucose, and therefore there is more brain activity in this. It's a highly spatially accurate technique, but the temporal resolution isn't as good as, say, EEG, which we'll talk about in a moment. But through this fMRI research, we've found that there are certain networks that the brain has intrinsically. So the default mode network is probably one you've heard about. It's responsible for the sort of self-referential processes that we go through. And it's found to be hyperactive in states of depression, for example. And a lot of the research has shown that psychedelics cause a decrease in within network functional connectivity. So that's the connectivity between these functionally similar regions. And especially with the default mode network, this self-referential network, which has been related to the sense of ego dissolution that you might experience when you're under the influence of a psychedelic. Psychedelics have also been shown to increase global integration of the brain and increase between network functional connectivity. So as much as the brain is separated into these networks, there's still crosstalk between these networks, and psychedelics enhance that crosstalk between these networks. Psychedelics also reduce the hierarchical sensory association cortex gradient. That's a bit of a mouthful. And essentially what that means is that when you're processing sensory information, it will generally go to the primary sensory areas, and it will be sent to the more integrative areas of the brain where it's all kind of pieced together. But when you take a psychedelic, it sort of flattens that hierarchy, which I think you might have mentioned in some of your lectures about the visual hierarchy. So essentially that might lead to these more synesthesic experiences that one might experience on psychedelics, or sort of piecing together information that wouldn't normally feel like it makes sense. Another leading theory in psychedelic neuroscience is the thalamocortical gating framework theory. And it's essentially that psychedelics increase the connectivity between the thalamus and the cortex, and the thalamus is thought to be a sensory gating region. And so if you're increasing the connectivity of the sensory gating region, perhaps you're letting more information flow, more sensory information into the cortex where it's processed. So this diagram is sort of speaking to the second point here, that there is an increase in between network functional connectivity. So imagine that this circle is the whole brain, and then you have the little circles which are just regions in the brain, and the different colors represent the different networks. And this is in the placebo condition. You know, there's some crosstalk between the different networks, but not so much. And then you give them a psychedelic, and suddenly everything is wiring together. Okay, then we have another technique of neuroimaging called EEG, electroencephalography. And this is a more direct recording of brain activity it's measuring the electrical activity. So with fMRI, we're just looking at changes in blood flow, making inferences about brain activity. With EEG, we're directly measuring the electrical activity of the neurons. And with in normal waking state, you'll have these 5 bands of frequency bands that generally the electrical activity will compartmentalize into. And generally, psychedelics cause this broadband desynchronization of all of these frequencies. Mostly with alpha, it will cause a decrease in the power, and alpha is associated with this relaxed state, and it's thought to be linked to this ego dissolution state with this reduction in alpha. There are also increases in the entropy of the signal diversity, and potentially this speaks to this more chaotic experience in the psychedelic state. There's also been shown to be increases in gamma after DMT, ayahuasca, and psilocybin, which potentially could explain this, an underlying mechanism of the mystical experience, as this gamma wave is responsible for awareness. But that is a bit more contentious. And so one of the leading frameworks in psychedelic neuroscience is the Rebus model. It stands for Relaxed Beliefs Under Psychedelics. And this is based on the predictive processing framework, which is very much a leading theory of consciousness. And it's the idea that we make beliefs, we have beliefs or priors about the world, and that's how it allows us to function quite efficiently. If you're constantly going around the world as a newborn baby in the dinosaur suit would be going around the world, you're having to learn all this new information. And luckily, we have these priors, so we don't have to consistently learn new things. Otherwise, life would be very, very chaotic. And so when you have these top-down predictions about the world, we have these sensory input that comes in. And if there's new input, it will lead to this prediction error, and then it will update the model. And so using that framework, it's thought that psychedelics sort of relax these priors, relax these beliefs about the world, and it allows for us to sort of receive more of this incoming sensory information and learn new things. And it's also thought that what often underpins psychiatric conditions are these maladaptive beliefs about the world. So if you're depressed, you might have these very highly precise, strong, and rigid beliefs that everything is crap, that you're a piece of poo, and depression can be very rigid and strong in its beliefs. And the same with addiction. You believe that the drug is very rewarding and that's the best thing ever. It's kind of a common theme potentially in many psychiatric conditions. And potentially under psychedelics, these priors might become more relaxed and allow you to have these new insights and perspectives into these potentially quite maladaptive beliefs that underpin these psychiatric conditions. And this has been seen in the clinical research. So this was a study conducted at Imperial College a fair few years ago, comparing the effects of psilocybin therapy to escitalopram. So it involved 2 psilocybin therapy sessions separated by 3 weeks, and this was the baseline resting state scan compared to 3 weeks after the last dose, so 6 weeks overall. And by at rest, I mean you get people to lie in the scanner and do nothing and think of nothing, and they're not on the drug. So this is 3 weeks after the experience. And this paper showed that there was a reduction in modularity. And modularity is the idea that the brain is separated into modules. And the more separated it is, the more sort of it's more rigid in its information processing. And for example, in depression, it's thought to be more modular. So this decrease in modularity, and it essentially means this increase in connectivity, is what is observed after psilocybin therapy, whereas there was no change after SSRIs, after escitalopram. And this was actually seen to be related to changes in depression scores with psilocybin over here. So potentially this increase in global integration is one of the mechanisms of psychedelic therapy. And yeah, this diagram is just broadly showing that. So with depression and addiction, for example, amongst other things, you'll have these deep troughs of fixed thinking that are quite rigid. It's quite difficult to get out of it. And then increased modularity, difficult for the information to transfer across the brain. And then during the trip, it flattens this energy landscape and allows information to move more freely and reduces the network segregation. And then after treatment, we have these persisting reductions in modularity and an increase in cognitive flexibility. And so people are able to escape from these negative thought loops. And so this increase in cognitive flexibility could potentially be reflecting these changes in beliefs. And this is seen in many, many different dimensions, as seen across several studies. We've seen increases in social functioning, agreeableness, spirituality, nature connection, extroversion, openness, well-being, sexual functioning, mindfulness. And all of these speak to the persisting effects of psychedelics. So this isn't just under the influence. Okay, so I'm going to speak a bit more now about what my main focus of research has been over the past while. This is a bit more of the clinical neuroscience angle. So across many psychiatric conditions, we have maladaptive reward function. So in substance use disorders, depression, anxiety, eating disorders, PTSD, we often find that there is dysfunction in reward circuitry. And so the reward circuitry, the primary reward circuitry in the brain is this mesocorticolimbic circuitry. So it's composing of this cortical prefrontal cortex region. We have the nucleus accumbens, which is linked to the reward and motivation aspects of things. The amygdala, which is the sort of more emotional processing, fear processing center. The ventral tegmental area, which is a real producer of dopamine, which is driving this reward. As well as the hippocampus, which is linked to memory. And together, this all works together, and it's thought to underpin. It's been shown that there's a lot of dysfunction in this circuitry in these disorders that I've listed here. And in the acute state, we have seen that this circuitry is directly impacted by psychedelics. And so we think potentially the impacts of psychedelics on this circuitry could also be influencing these clinical benefits that we're seeing in clinical populations. So we've seen that LSD acutely increases hedonic, motivational, and affective processing of feedback. So that's in the acute state. We've also seen this with DMT. So DMT has also decreased connectivity between the nucleus accumbens and the VTA. So these regions in the sort of disordered state, they'll have they'll be strongly connected. So this reduction in connectivity with DMT could be a clinical mechanism potentially, and also increased connectivity between the anterior cingulate cortex with this nucleus accumbens and the medial prefrontal cortex. So these more sort of higher order cortical regions having influence on this perhaps disordered nucleus accumbens, which is responsible for driving this disorder, this dysfunctional reward system. We've also seen at rest that psilocybin acutely decreases amygdala connectivity, and this amygdala is responsible for, as I said, emotional and fear processing. So this reduction in connectivity could potentially be what is helping people access these emotional states for things they might normally find challenging in their normal waking consciousness. And this has been shown not just at rest, so not just doing nothing, but people shown psilocybin in the people that have taken psilocybin in the scanner when they are shown some images of emotional faces. That's a commonly used paradigm in this sort of field of research. The amygdala reactivity to negative stimuli was lower after psilocybin administration. And this has also been seen with LSD. And again, just reiterating that this could potentially indicate altered emotional processing in these states, which could contribute to this increased emotional openness. And this has been seen to have persisting effects on amygdala activity. And so in Lior's research several years ago, they showed that one day after the psilocybin experience, there was an increase in reactivity, which is quite unusual because you think in the acute state, there was this decrease in amygdala activity. But then the next day there's sort of an uptick, and then other studies have shown there is a decrease in the weeks, in like up to a week after, potentially 1 month after. So potentially there's this dip, and it increases, and then it normalizes into a more healthy state in the depressed or addicted or whichever brain. And so this is a piece of work I've been working on recently. I've been looking into these areas of the brain and seeing how the connectivity of these regions has been altered. So this again was from the study comparing escitalopram to psilocybin, and essentially, again, reiterating that they're not under the influence of these drugs. This is 3 weeks after. I looked at the amygdala, nucleus accumbens, the areas of the mesocortical limbic circuitry that I've showed you, and demonstrated that psilocybin therapy mediates these increases in the within and between connectivity of the limbic stratum. So that is the nucleus accumbens with hubs in the salient network and more prefrontal regions. And that speaks to this increase in healthy functioning of the reward system. Whereas with escitalopram, we saw reductions in the connectivity of this. And potentially with escitalopram, a lot of people report feeling emotionally numb. And so we've kind of inferred that potentially this could be a mechanism that differentiates the two different drugs. Psilocybin therapy enhancing ability to feel emotions, and escitalopram reducing that network connectivity, which also leads to these that dampening effect. We also showed this with a paper that Leo and I published. This is from my master's thesis, comparing these two drugs, these two treatments for depression. And we looked into several different levels. So we asked them about their feelings of anhedonia, and anhedonia is essentially the inability to feel pleasure, and it's a symptom commonly reported in depression. And we showed that psilocybin outperforms escitalopram at treating symptoms of anhedonia. We also got them to listen to a piece of music in the fMRI scanner, and we got them during the scan to, so they listened to the music in the scanner, they came out of the scanner and tracked their experiences of valence and arousal, so how much pleasure they felt from the music. And we also asked them about their overall subjective experience of music. And we showed that following psilocybin therapy, there were increases in these feelings of vitality in response to music, so that's the overall experience, whereas there were decreases in escitalopram. And again, on this sort of more continuous basis, where we asked them to track their feelings of valence and arousal, we looked at this part of the music called musical surprises, which are linked to the rewarding aspect of music. It's the rewarding elements in the musical piece. And we showed that escitalopram again reduced that surprise evoked valence, which psilocybin didn't cause a change in. So that kind of goes to show on the behavioural and subjective level that escitalopram is blunting these feelings of reward, whereas psilocybin is enhancing or maintaining them. And from our work with fMRI, we also showed that psilocybin caused a decrease in the activation of this ventral medial prefrontal cortex and angular gyrus, which are regions in this default mode network, but there was greater activation of the sensory regions. So potentially that is speaking to this decrease in activity in the default mode network, which is allowing you to feel more and to process these sensory processes, but also this rewarding aspect of music in this situation. Okay, so this is the project I'm currently working on. It looks very complex here, but I'll talk you through it now. At UCL, we're doing the Unity project, and Unity stands for Understanding Neuroplasticity Induced by Tryptamines. And the main question we're asking is, can psychedelics help people improve their drinking habits, and how? So we've had 40 participants so far, and we're looking for 120 heavy drinkers. So not fully, he's just put his hand up at the back. And they will receive either one of 4 interventions, with one of them being 25 milligrams of NN-DMT in the intravenous infusion over 10 minutes, a very big dose breakthrough experience. And this is in a memory reconsolidation interference paradigm. So memory reconsolidation is the idea that memory, sort of maladaptive reward memories, are what underpins a lot of psychiatric conditions. In this context, if you see an alcoholic drink, you'll have a reward memory associated with that, and then the memory will become activated, and in that process of reconsolidation, you can interfere with that reward memory and rewrite that memory essentially. So that is the main hypothesis of my supervisor, Ravi Das. But my main primary focus is looking at the fMRI from before to after. So we do an fMRI scan at baseline, which is 1 week before, potentially DMT or one of the other interventions. And then 2 weeks after their, whichever experience they've had, then we're looking at resting state, so doing nothing in the scanner. We're showing them pictures of beer to see, or whichever drink they like, to see how their brain responds to it. And we're also showing them movies associated with drinking. Although 2 films actually have Tom Cruise in it, so we're actually going to look at the Tom Cruise effect in the brain, the anti-effects of that. But yeah, we're doing we're collecting a lot of other measures. We're collecting cognitive measures, we're collecting questionnaires, we're collecting EMA, so ecological momentary assessment, where we get people to give us a little voice note of how their day is going and looking at changes in natural language processing. So this is very much an ongoing study. It's estimated to end by March 2027. That's when we have to complete all of our endpoints up to the 9-month period. And broadly, we want to develop an open source neuroimaging database to investigate mechanisms linked to these persisting and subacute effects of psychedelics. We also want to detect this functional neuroplasticity changes following psychedelics, observed through these large-scale cortical networks and this reward circuitry. And also we want to identify whether these network changes are predictive of individual phenotypes and changes in drinking and well-being. Yeah, I can't report on any of the data as of yet because the study is still ongoing. If you would like to take part, then please go to our website. I'll put it on at the end, or if you want to come chat with me at the end. I'll speak more about the rest of our lab's work as well. But back to sort of summarize what we've been talking about and think about some of the questions that still remain in this field. One of the big ones is how we bridge the gap between these different layers. It's all well and good sort of discussing them at the different in the different disciplines, but actually creating a cohesive theory of how they all work together. And that's an issue not just in psychedelic neuroscience, but more broadly, this translational gap. How do you take it from the head twitching rat to the human? Also, probably what you've been thinking this whole time, I've just rattled on about how all these brain regions are disordered and what have you, but psychiatric conditions are not just a biological disorder, it's a biopsychosocial issue. So I've just spoken about a few of these parts of this biological circle here today, but there are the psychological components and the social and environmental components. But I also think that this is where psychedelics are really helpful and kind of reiterate that this model is definitely what we need to actually think about. It kind of challenges this classic biological psychiatry framework, because as this, within the psychedelic experience, it's essentially considered as a catalyst for change, and it's in combination with this set, so the preparation, the expectation, the surroundings that you're in, the mindset that you're in, as well as the setting, which is the physical environment, a sort of social and cultural environment. Is these factors with this biological basis up here, with these brain dynamics, with this connectedness and interpersonal functioning, the emotion regulation, the cognition, and all the other factors with psychotherapy that leads to these positive outcomes with psychedelics. The biological basis is just one of the parts of it. And I think that this is where psychedelics are really interesting in psychiatry, because it challenges this take a pill, go home, and that will fix you sort of mindset. And I think this is where a lot of the challenges have come from bringing it to the MHRA, to the FDA, because they've not understood that framework before. So it's kind of challenged the entire system in psychiatry. But one of the other questions in this field I find really interesting is whether the psychedelic effects are actually really necessary. I'm more on this side where I believe that it's a behavioural catalyst, and that the experience gained in the psychedelic trip and the psychological insights are what is central to producing these long-term changes in behaviour. But there's this other camp which believe in this biological model. And it's really, really interesting work from David Olson's lab, who they've developed this drug called tabenanthalog, and it's essentially this non-psychedelic psychedelic. And they developed it's been seen in mice models to show that there is an increase in neuroplasticity in the same way that psychedelics do, which leads to these therapeutic effects. I think there's soon to be introducing it in a phase 1 study in humans, or it might currently be in that study. But if you can remove the psychedelic effect and get these therapeutic benefits, potentially this could be a game changer in psychiatry. I mean, you're taking the fun out of the trip, I suppose, but for some, the trip, it might be undesirable. And so if it is just this increase in neuroplasticity, it could really be a game changer. But yeah, it's an interesting question. I don't know if maybe we can talk about it at the end, if any of you have any strong opinions about it. No one else. Fine. Other questions that we have is, how do you double blind psychedelics? And I guess that's linked to another question we have here of what is the role of expectancy? If you expect that you're going to be healed after this psychedelic experience, especially after reading all of these news headlines, you might expect that you're going to have this life-changing experience and it's going to help you. Perhaps that is what is driving these positive clinical outcomes. But also in terms of rigorous science, double blinding a psychedelic experience, both on the behalf of the participant who's consuming the drug, as well as the researcher who can observe the participant, it means that you can kind of tell if they're on it. Does that, normally in science, you need a placebo to have this unbiased comparison. Also, there are other mechanisms of psychedelics in that sort of biopsychosocial diagram that I showed you. Some of them were on there, but there is evidence that there are other mechanisms, such as it has anti-inflammatory properties, which potentially could underpin some of its clinical efficacy. So in cases of depression, for example, it's been shown that there is an increase in inflammation, neuroinflammation, and potentially this reduction in inflammation with psychedelics could be one of the mechanisms. Perhaps it's just the whole package. But how do you study the whole package is one of the questions. And so to conclude, the therapeutic action of psychedelics spans from molecular alterations to network changes to observable behaviour to the social aspects to the psychological aspects. It's sort of the whole package deal. With psychedelics, there is an enhancement in neuroplasticity, which may manifest as a reopening of this critical period, a temporary window for accelerated learning and unlearning, as well as the clinical benefits potentially are driven by the reorganization of these brain networks and the normalization of maladaptive reward system function. And this biological reset could promote the cognitive flexibility by allowing a reduction in these rigid priors. And again, reiterating that psychedelics offer this transdiagnostic mechanism for many disorders, and it strongly validates this biopsychosocial model of psychiatric care. So with that, I'd like to thank the rest of my lab at UCL. You can go to our website here to learn more about our research. My supervisors, Jeremy and Ravi. We are doing a lot more research we've got a psilocybin study on at the moment. We're going to be starting an MDMA one, LSD one led by Anjali, lots of nitrous oxide work, pretty much any drug do research. But yeah, if you'd like to know more about what we're doing, please head over to our website or come chat. Yeah, if you have any questions, please feel free.